Hi, everybody. Uh, we have Michele Mansfield here. She joined Penn State Grozinger Lab as part of the Center for Pollinator Research in April of 2021 to help develop the Honey and Pollen Diagnostic Lab. A plant pathologist by trade, she's truly come to appreciate the world of pollen and pollinators. Her main professional interest is in diagnostic method development. She also serves as an advisory plant pathologist for a startup PSU, Penn State University, alumni-based ag technology company, and is an avid gardener, which I also just learned today. That's very cool. Without further ado, Michele, why don't you take it away?
All right, thank you, Carissa. It's fun to see. It's fun to like meet, have emails and faces and like get to know some personalities this evening. Yeah. So as Carissa said, I joined the lab in 2021. And when I started, I didn't know anything about beekeeping. I was hired to be a diagnostician. So it was like and I have to say, like, beekeepers have been one of the most interesting and fun and engaging group of people I've worked with. Um, Usually I was telling people as a plant pathologist like what was killing their things. So they weren't always like in the, they weren't like, it wasn't the top of their game to be like, cheerful and happy about things. So for the most part, this has been a real joy to work with you folks. Just as a quick overview, I'm going to go over like what the lab offers currently. We have Pollen ID, which is identification of pollen and pollen samples and honey samples. And you'll also hear me refer to this as pollen metabarcoding, which is a mouthful. So I just usually say pollen ID service because I get sick of saying it too. We also have the spotted lanternfly detection service. That's the newer of the two services. And at the end of my talk, I'll talk about how I hope to expand some of the services as well. One of the other things I want to focus on is more of how are the techniques at the Honey and Pollen Diagnostic Lab that we use to help research projects. And some of you may have been involved in Dr. Michelle Lee's. I know there's a lot of Michelles on this talk. We used to say, like, I said she was the more fun version because she gets the double L's and she's younger than me. So I was like, more energy. But I'm going to talk a little bit about her project and just some kind of the neat things she found out about from samples from Pennsylvania, maybe from some of you folks. And then I'll also really briefly go over some of the other resources from the Penn State Center for Pollinator Research. Just in case, you probably have heard many of these if you've heard talks from other people and Dr. Grozinger Labs or some of the other folks in the center. Okay, so some of you may know this, some of you may not. Traditionally, when you're doing assignment of plant sources and honey samples or pollen, you were just identifying the pollen. To date, we still don't have a way to characterize nectar in its own right. DNA is a unique character identifier for most organisms, which is why you use pollen identification to figure out what plants, may be making up your honey. And what I do tell people is that no, you can't necessarily strictly attribute nectar sources directly to the pollen that's in there. You should look at it more as of what have they foraged on. So it's more of a foraging fingerprint necessarily than a perfect nectar fingerprint. So just keep that in mind. But right now it's the only technology we have. In the past, traditionally pollen identification was done by, and I'm not gonna lie, these people are artists, okay? Being able to look under a microscope and literally filter out, count, and identify each individual pollen grain. They grid it out by physical pale. They use your physical characteristics, and pollen sometimes doesn't have that many physical characteristics. I mean, I see this like sweet, like electrified picture. By the way, I will say my 8-year-old found this picture for me. I do not do amazing pictures of this of pollen, but it requires extensive training to do this. It's also, this is one of my favorite words, say this fast a couple times or after a few drinks, Melissa Paleontology. I do work with a Melissa Paleontologist. She is a wonderful woman, but it does require extensive training. It's not high throughput in the sense that you can run through hundreds of samples in, a couple month or weekly period. The resolutions generally at the family level, although really, really good. My cross-class mesopaleontologists, they can get down to the usually the family level, sometimes the genus level, but it's also very time consuming because in a lot of respects, this is almost more art form than it is just pure science. But it is also that. I like to think that we work together. We are not in conflict for one another. They both that give insight in different ways. Okay. All right. So just to, I know like everyone, I say I really hate making people think about things like math or science after 4 p.m., but I promise this won't be painful. So metabarcoding is a fancy way, but when I was old school, I used to refer to this as just sequencing a piece of DNA to identify an organism. Okay, so metabarcoding is kind of fancy. When you have traditional, what they call DNA barcoding or sequencing, if any of you are familiar with that term, you have to have an organism either by itself as a pure sample or a pure isolate of something or pure tissue or whatever it is that you're looking for. And then that one organism is taken through the steps, and then you take a piece of DNA that identifies it, you plug it in against a database, and it tells you what the closest match is. Metabarcoding, on the other hand, you don't need to have a pure isolate of something. You can take an environmental sample. You can take soil. You can take a pollen trap sample, which is not made up of one thing. It's made of many, many different kinds of plants, usually. And then after you go through that process, you can identify everything in one shot. So instead of basically cutting a picture out and then trying to put it back together by taking a sequence of each individual organism, you can take a snapshot of everybody at once. So here's kind of my analogy. Imagine if we were all in a room together and everyone was out in the hallway and I was standing in the doorway. Each one of you would come in and I would shake your hand and I would identify you one at a time. Now imagine that you're in a big building. Like let's take Beaver Stadium, for example. I have spent a lot of time there since my husband works for athletics. Imagine that I'm standing at the top of Beaver Stadium and I can see in the whole stadium. I can take a snapshot and I can identify everybody in one shot. It's an extremely powerful technique. It gives us insight not only to what organisms are present, but how they might be interacting with one another. And in this case, not only can I tell you what pollen is in your sample, I can tell you the percentage overall of what that pollen makes up of, or what that particular plant makes up in that sample. Okay, now to the nitty gritty. It gets easier from here. If you're interested in sending samples in, I do have the QR code here. You can either email me directly, I have my contact information, or you can go to the QR code, visit the website. I strongly encourage everyone to read over the FAQs on the website for the services too. There might be details that I don't cover in my talk that might be of interest to you. So I take samples from anywhere in the world from anyone who wants to send them. One of the funnier ones I've gotten lately is somebody literally took a piece of tape, stuck it to their car because they wanted to know what pollens were in their area that were making them sneeze. I'm like, I can't guarantee I can tell you which one's actually your problem, but I can't identify what's on the tape for you. It was pretty hilarious. They were quite happy. But again, this is for anyone who wants to send samples. The lab accepts honey, either uncapped honey, that's commercial ready, or as you can see here at comb scraping. Comb scraping samples do tend to have the most informative amount of info. They're the most informative in terms of even light screening or the extraction process itself will remove some pollen. Now we can still get information out of a commercial ready or an unkept sample. It's just occasionally I tell people if they're not quite pleased with what they've gotten in their results, doing a comb scraping might be more the way to go. We also take pollen from pollen traps, and I will take pieces of tape. I will also I've gotten from many researchers will send me little tiny bees that we gently remove. Now the bees aren't alive anymore, but we gently remove the pollen, and usually I like to send the insect back to them in case they want to use it for a collection. But we take anything that will take or that has pollen on it. I'm hoping I work with a group in Bolivia that wants to send me pollen that they have very gently removed from hummingbirds with a toothbrush. I think that would be kind of exciting. Okay, Carissa can tell you all about this too. But when you get in touch with me, there's a submission form on our website, and after, I go through and I make sure I have all your information and you let me know what kind of sample that you plan on sending, whether it's pollen or honey. We send a collection kit. The collection kits are good because they provide a standard of uniformity so that we know we have enough sample. Like if a tube breaks in transit, which I haven't had that happen, but I always ask for two just in case. The sample will come for 2 tubes for honey, 1 tube for pollen. And no, you don't need to fill it up for pollen. Usually I tell people get to the first, like the 5 mark on the 5 ml tube. That's usually enough. There's information. I ask you to fill out information on your sample. Most of this information is actually so that when I send you your report back, you remember, you make sure that I have all the information correct and that you can identify your sample when you get it back, especially if you submit more than one sample. There's a return envelope. Postage is already paid for when you've paid for the sample. And everything is kind of an all-inclusive kind of deal. And I always tell people, please ask me if you're not sure about the directions. I'm not a beekeeper, so when the first round of directions came out, I had a lot of questions like, lady, what are you talking about? I've gotten much better at like the directions now, and I think that they're a lot more palatable to most people. Okay. Again, 2 tubes for sample. We'll take commercial ready honey or uncapped sample, which is a comb scraping. Comb scrapings can be beneficial as well because if you know when a particular frame was filled, it might give you a better snapshot. Although occasionally a lot of beekeepers, they want to know collectively what the bees have been visiting throughout the season, which is why they might prefer like an extracted sample. Once the samples get back at the lab, people are always worried that they're going to send me, like if they send me a sample that has wax in it, that's going to be a problem. It's not a problem. I take it back to the lab, I warm it up in an incubator, and essentially what I'm doing is I'm taking a fun little centrifuge and like basically creating a little ball at the bottom of the tube of the pollen. We pull it out of the honey. And that big sample that you gave me now is an itty bitty, about a 1 ml sample. So we reduce everything to get down to the pollen. That's the goal here. Pollen traps. This is kind of like more if you collected samples for Michelle Lee's project. Excuse me. This is kind of more the format you would have followed. But again, if you don't want to starve your bees, I get it. If you just want to be able to collect it for a day and send me a quick snapshot, that's almost plenty of honey, or excuse me, pollen for a collection to find out what you were probably after. So again, standard collected for 7 days. You hand mix it, send it in the tube. That's for if you can't send me very much. Fill it all the way to the top if you can. If you can. If you cannot, it'll work. And then the sample is homogenized so that we take all that pollen. We make sure that when I take the sample to get after the DNA, that all the pollen that's in the sample is represented equally. That way we can make sure our proportions are correct at the end when I report it for you. But a pollinator, I've literally taken itty bitty bees and sometimes we vacuumed it. This vacuum is hilariously tiny, but it's the same. The idea is the same. You want to concentrate and get to the pollen. All right. Okay, I know details and science. I promise I won't hurt your head though. I do. My kids have even sat through this more than once. So really what I'm after when I get a sample is to get to the pollen and then break that pollen open to get to the DNA. And believe it or not, pollen is some of the toughest stuff I've ever worked on, and that includes like tree bark and like a lot of other really bizarre things. This is a bead mill. So the tube that we use has little chrome beads, and literally this thing shakes the crap out of a sample and breaks it apart. My daughter loves this instrument. It's called a bead mill because it's extremely loud and violent, and I'm like, it's the noisiest thing in my lab easily. Once we have that pollen beaten up, it's gone through, I use a commercial DNA extraction kit that will then fully break up the sample, isolate the DNA, and then also purify the DNA because we want things to be as pure as possible. And also, I sometimes have to adjust the concentration. The idea being that if all the samples are on kind of the same like level playing field, we know we're going to be able to provide the most accurate results. So the most complicated part of this entire operation is called PCR. PCR is where you're literally photocopying a piece of DNA of interest. The piece of DNA we use in this whole procedure is one that's very characteristic of being able to identify a plant to genus level. This particular piece of DNA has been sequenced worldwide for 20 years or more. So there's a very good, very level database, lots of information out there that we can use to make sure, again, we have the most accurate results. Again, the PCR is photocopying to make more of it. Then that DNA is then labeled. Literally, it's labeled throughout this PCR photocopying process. And then finally, it's sequenced, which is what will give you the actual data that you're after for identification. All right. So, what happens after all that magic? Well, essentially, you get a sample report, and this is kind of what you would expect to see if you send me a sample. We provide a lot of the information. Again, this is all information you've sent me. And I've had people be like, oh no, like I think I sent you something incorrect, or it's good. That way we all make sure that we fact check one another. I mean, I'm human and I don't ever, my data entry skills are usually pretty on par, but every once in a while. All right, so it provides information here, and then really the meat and potatoes of what you're interested in is it will to give you a pie chart that basically will tell you what plants to the genus level were found in your sample and at what proportions. So here we've got Baneberry. This is a pretty typical, I had to guess, you should think I would know this. This probably looks like a late spring sample if I had to guess on this one. But again, like we provide not every service will give you anything below 5%. I try to make sure that you get everything, even things that are less than 1%. Okay. So I know changing gears here really quick. So the other service that the lab offers now, and this is kind of funny because this came about in the weirdest way possible. I had just started working in the lab and I was kind of just wrapping my heads around getting the Metabar, the pollen identification service up and running. And I kept getting all these questions about spotter and lanternfly. I'm like, what is this? Like, why are people so worried about this? Well, it turns out it's all over the news, and now we all know why everyone's worried about it. And I had quite a few researchers, including Dr. Robin Underwood, who some of you may be familiar with, because she was getting a lot of questions like, hey, like spotter and lanternfly, like what's going on here? And turns out that lab at Rutgers actually had a method to detect spotted lanternfly DNA off of surfaces. Like they literally would wipe like stop signs, they'd sample water from like all kinds of environmental samples. And I was like, huh, I wonder, I bet we could get this to work with honey. And with a little bit of tinkering, a little bit of help from the lab, that their lab as well, turns out we can detect spotter and lanternfly in honey. And basically, what happens if you're not familiar with the process, spotter and lanternflies are basically gigantic aphids. They suck on like on a plant, usually a tree. They suck the sap out, and because they're mostly sap feeders, they basically, I want to, for lack of a better word, pee. They exude out sugar water. And bees, as you probably know, will go after many a different type of sugar source. And if you ever seen a spotter lanternfly, it actually was really funny too. I had to get all type of like, special permission to work with spotter lanternflies like 3 or 4 years ago. Literally, I find them dead outside of my building come like August. I'm like, oh, I'm glad I had to do all that paperwork. But anyway, we offer this service now because a lot of beekeepers want to know whether or not they have spotted lanternfly or honeydew in their honey. Another thing that keyed us into the fact that lanternflies have some type of effect on honey is the fact that we were ending up with plants and honey samples that the sequencing was fine. There wasn't a mistake. The database wasn't making a miscall, but we had plants that didn't make sense. Like the bloom time shouldn't have been happening when these particular plants were showing up. And we now are pretty sure that it's showing up via the exudate from the spotter lanternfly. So what this is kind of showing you here, and the process is very much the same as if you send me a honey sample, I'm going to be doing the same thing with the DNA extraction, the whole 9 yards with the lanternfly that I would be doing with the pollen ID. And I'm doing another type of fancy DNA replication. And essentially what this is showing you is with each round of photocopying of the DNA, that's what these number are down here. It's called cycles. So imagine a photocopier spitting out like, however many copies you're at. The more lanternfly DNA you have in a sample, the sooner it's going to start showing up. So, and it's kind of odd, and you wouldn't necessarily think about it this way, but the lower the number means the more spotted lanternfly DNA is present in a sample. Now, that doesn't really have any bearing on whether it's one spotted lanternfly or five that contributed, because there's really no way to know that. But it gives an idea of like, how much honeydew might be contributing to the honey overall. And again, I kind of leave Robin Underwood to talk about whether or not having lanternfly honeydew is good or not in your honey. I do know that there have been some reports that it has higher medicinal value, so maybe it's not all bad. But again, this is another service we offer. We're hoping this year I love the university. I love working at Penn State, but I'm not going to lie, there's a lot of red tapes to go through in accounting to get new services up and running. We're hoping to offer a pollen nutritional assessment, specifically protein lipid ratios of pollen, potentially total carbohydrates. A lot of beekeepers are interested in knowing essentially how beneficial their pollen is for the pollen that their bees are collecting, and whether or not that may be impacting their hive health in any way. We're also hoping to offer some sugar profiles for honey, glucose, sucrose, fructose, um, amylase, diastase, and vertase activity. That's mostly freshness parameters, as well as, it's the basic things like pH, Brix content, and water content. Contaminant detection, I haven't gotten there yet. Mostly I refer people to go to Cornell if you're looking for pesticide residue or things like that, but there has been a lot of discussion about like whether you can test for adulteration in honey. I'm not in that business yet. But again, it's on my radar to think about. All right, so now here's like the kind of fun application portion of now that you've had to listen to how I do things, now you can listen to like what it's actually used for. Okay. So the pollen meta bar cutting, pollen ID, what are the applications? I mean, it might make sense to you, it's common sense, but not necessarily, maybe not. So the Center for Pollinator Research isn't just focused on honeybees, we're focused on all wild bees and all pollinators. The more we know about what they're visiting, what they're foraging on, the better we know how to take care of them. Obviously, as a beekeeper, you want to know what your bees visit and whether or not that has an impact on the honey quality, hive health, things like that. And again, overall, like the overarching idea is to drive conservation efforts, especially for threatened species or even species that are threatened in that particular area where they were once abundant. All right, so I'm going to introduce Dr. Michele Lee, like kind of via this picture here. I first met Michele in 2022 because she sent me a boatload of samples when she was finishing up her PhD in Santa Barbara. Now these were bumblebee samples and wild bee samples, and what they were looking at was if they could basically see and observe what the honeybee, or excuse me, the bumblebees were foraging on, and whether or not like the metabarcoding would give any type of like extra insight to what they were seeing. And turns out the metabarcoding, you can just see here on the side, significantly more genera of plants were identified that weren't even the bees weren't even observed visiting. So it kind of gives an insight that, as much as we try to do a great job by visually observing insects in the field, sometimes like we're human, right? I can't fly around with the bee, like you want to let them do their job too. So that was kind of like a fun first application to using the metabarcoding for a major project. Also, Michele and she started at Penn State in 2023 as a postdoc, and I worked more closely with her. We had an idea of, hey, I wonder if we could start looking at floral resource timing, which actually will lead into the pollen collection that she did for many of you in 2024. So Penn State has a huge collection of pollinators in the Frost Museum, and we were interested about wonder if we could get, viable like pollen that still was good enough to be sequenced. We could get that off of samples that are, archived or for are ancient. So basically went through the whole Frost Museum and pulled out the good samples and kind of mapped out, when and where we were seeing certain plants. It gives you an interesting historic perspective of how things have changed over time. Okay. And then again, like I said, floral resource timing, this is kind of a little mini study that she started with looking at what types of plants are we seeing throughout the year? Like, when does it matter? What matter most for pollinators, including honeybees? Okay. Oh, I skipped past. Uh-oh. Okay. All right. I think I'm back where I need to be. Let me check. Yes. Okay. My finger was a little trigger happy on that one. Okay, so spring 2024, this is Dr. Michele Lee's work. This was a project where she looked at pollen from beekeepers from 6 regions: York, Philadelphia, and Pittsburgh. The sample collection started in March, and I believe like she went and made sure everyone had pollen traps. These were like the commercial like Better Bee or whatever beekeeping supply company offers like the wood pollen traps. The collection went from March to June, and it was a total of 284 pollen samples. That the lab I was real busy that summer, real busy. In fact, it actually like got me to hire some additional help because for a while it was just me, myself, and I. The goal of the project was to look at how urban landscapes affect bloom time and pollen quality and quantity in terms of what plants were found in them. And again, I will say, I let Carissa know this too. I just got these slides this morning, so if I'm a little awkward on describing what's going on each slide, I practice, but I'm still kind of learning through this data just a bit. Okay, so essentially what this slide is showing is what she means by impervious essentially means impervious means it's not it's the concrete jungle, right? So the brighter the color, the more urban density is going to be likely in a particular location. And this is kind of an idea of a general map of where the apiaries were located, where pollen was collected. And this is just kind of set the team. So she was focusing on fairly urban dense areas. And again, keep in mind as we're looking, impervious more or less means like concrete jungle. So more impervious, more concrete. That's kind of how I went through this. I know I told her I felt bad that I was dummying down her talk. Okay. So what does this mean from the data that we got from the pollen identification in the samples? So essentially what she was seeing, and this is kind of just an overall arching, I guess, you know, trend that she saw over all three general regions. The higher the impervious surface, so the higher like the more urban or more concrete it was, she noticed that there was zero point 0.6 increase of the number of days of bloom time was earlier. So I'm sure many of you have noticed this. I live in a little town where my magnolia tree in the backyard is at least 2 weeks behind the magnolia tree that's less than a mile away, but in downtown. But yeah, it's amazing. Like, so she started to notice this trend, and now she's wondering, okay, all right, well, that's something we might expect. And this was true across all three different locations. And now when you're looking specifically at the species of plants that were found. So I'm going to give you guys a quick rundown. Acer is maple, Fraxinus is ash, Malus would be apple, Prunus plum, plum or cherry, Pyrus would be pear, Quercus is one of my favorites. That would be oak, and then Salix, which is willow tree. What she was noticing that as you're looking across these sites, that it was pretty consistent across tree genre that the bloom time was much earlier in all these locations when you looked at how impervious the surface was. Okay, so now when you're looking at, and that was for trees specifically. So the pattern, interestingly, wasn't as obvious or noticeable in herbaceous trees. So it would be like, not trees, not, and like nothing, like shrubs kind of fall in that in between area. Brassica would be kind of like your mustards that you'll see in early spring. Silastris is bittersweet. Rosa, that one's pretty obvious. Rubus would be caneberries, so blackberries, raspberries, Taraxacum, everyone's favorite thing that pops up in your yard in spring. Dandelions, Toxicodendron, my least favorite, that would be poison ivy. Bees love poison ivy, it's nuts. Trifolium clover and viburnum. Again, the trend wasn't as obvious across these guys, but it was still noticeable. So again, earlier bloom time, later bloom time. We're noticing us across all the regions that we're seeing that obviously more urbanization, a slightly earlier bloom time. And we're still trying to figure out what this means. So essentially, like, at least at this point with where the data analysis is, there appears to be what she's calling an urban heat island effect. So for every 10% increase in the concrete jungle, so more urban density, there's not quite, but there's an increase in the mean temperature. Now, this might not look like a lot, but over time, this means a lot to a plant. They can't get away, they can't escape. So overall, and that's actually a pretty high number. And again, interestingly, she saw it more on minimum temperatures than maximum temperatures. So it almost seems like once you get to a certain temperature, you're not seeing that the urban effect is making things drastically warmer, but you will see an effect when you're at the lower end of the temperature range. This data is still currently being worked on. She just completed her first year as a full-time teaching faculty at the Utah State, and she's now excited to revisit it, and she wishes she could give more information herself. She turned it over to me and trusted me. But hopefully you will be hearing from her at some point, because we're continuing to do the nutrition evaluation, so protein, lipid, carbohydrate analysis. Because it's not just, okay, we know that the bloom time is earlier. We know these species are showing up earlier than expected. But what does that mean about the overall nutrition of the pollen itself? So hopefully that will be coming soon. Okay. All right. So I'm going to go through just a few little things that talk about like some of the other resources that are at the Center of Pollinator Research. This is the QR code for the overall center's website. And there's tons of really good resources, specifically Pennsylvania pollinators, what types of plants you can put in your yard and where you can find them. If you want to know a little more, probably better detail than I'm providing about some of the research. And then again, just, kind of like how we're diversified across the research center. If you teach or know a teacher or want to get your kids into some stuff, there's some excellent information here about the class materials. And then we do have a research library where I'll talk about here in a second. Okay. Some of you may or may not be familiar with Beescape. This is a great tool if you're interested in knowing. This is actually kind of funny because like let's see, I live like kind of like up here, like off the map in this area, but it's fun to see how landscape is utilized. It's a good thing to know because it definitely can impact of when you think about like if I do I want to add more colonies, do I want to do I want to expand, or do I want to tone down, like what do I want to do? But think about how the land is managed and what it could might mean for bees in general or pollinators in general. And sometimes it's just kind of fun to see like what's around your area that you get more of a bird's eye view. Okay. All right, and then this is kind of I absolutely love this is an additional resource that I had some input on. So for full disclosure, one of the reasons I did not learn how to identify pollen under a microscope terribly well is I have about 2 minutes of looking at something under a microscope before I need to Dramamine. I won't go any farther with that, but yes, people get car sick. I get microscope sick, which is one of the major driving forces of why I ended up being a molecular diagnostician. Okay, but what I will say is what I love, this is an additional resource. This is free. So many beekeepers want to try to identify what pollen is in their honey and pollen samples on their own. And this is great. Microscopes have come down significantly in cost. You don't need a great one. I use my 8-year-old's microscope and it works just fine. I mean, I do have her find the pollen first and I look at it when it's stationary. But what this does is if you have an idea of what plants are in your area or you want to collect pollen from those plants and look it under a microscope and you know what it is, you can get a good idea of what that plant is. You can identify it just by looking, okay, there's a plant. And these also have high resolution, high quality microscopic images of the pollen. Right now, we have everything the Arboretum, of course, covered, but a significant number, I'm really pushing to get a significant number of what people would consider weeds, invasives, but are still around, something you might actually see. I'm trying to expand this, but this is a great resource and you can feel free to ask me about that at some point. Okay. And as of that, like I will take any questions. And hopefully I didn't bore the pants off you guys.
Heck
That's the
no.
problem with Zoom. When I'm in a room full of people, I can like read the room and read the mood.
Yeah, I know. I know. And I think most people have their cameras off anyway. So,
I would if
um,
I honestly I would too if I weren't the one presenting.
So we have one question in the chat. Michael says, with a pollen sample, my thing is hovering over it. I can't, it won't move. I can't read it now.
It
Um,
doesn't want to play nice with the scrolling.
ah,
Chris, I
okay.
can see it. Do you want me to read it?
Yes, can you read it? I can't. Thank you.
Yes. Okay. Michael says, with a pollen sample, there will be pollen from many different sources. You then said you take a slice of pollen up to the sections. Where are my glasses? There we go. Do those sections correspond to certain genomes? Are you using common section of DNA from each different pollen type to make the identification?
Okay, so we use basically I think I'm understanding your question here. The pollen sample as a whole is taken and homogenized, so everybody's represented. So when I take my subsection that I take to do the DNA extraction, the idea is that hopefully you're equally representing everyone. But the specific marker is called ITS1 and ITS2. These are extremely common. They're known. It's been a known plant identifier for almost 20 years. But what we do is once that section of DNA is caught and captured, it pushes it against the database, and a lot of I at least know two other groups that offer pollen identification this way. They only either use ITS1 or ITS2, or they use another genomic identifier. Those don't have the resolution if you're not using them together. We use two. So we use two different sections of DNA, which makes it more powerful. And in theory, we could say get to species level, but I'm hesitant to do that primarily just because it's the length of the sequence that matters. And with the current technology of sequencing, you can only get to a certain size. I'm hoping this improves. Hoping like next year I can get to species resolution. I'm not sure if I answered your question, but the idea being that if the homogenization has been on point and everything is and the DNA is normalized so everyone's on a level playing field, the representation should be fairly accurate of the proportions.
Great. He says that was helpful. Anybody else have a question? You can unmute and ask.
I do. I'm typing it.
Okay. I have a question too, so I'll wait for you, Donna.
Why don't you go ahead?
Okay. Um, I mean, okay. So, Michelle, you had talked about the other services that you're trying to get set up.
Yeah.
What and you did mention, I can't remember what the glucose, fructose, sucrose, and then the other ones that the parameters I can't remember that would determine the age of the honey, I think it was.
Yeah, that one. I just acquired information about a kit that will do that. It's the excuse me, amylase. It's an amylase dietase kit.
Oh, okay, yeah.
And it's more like a freshness evaluator. It can also tell you if the honey isn't being stored correctly, like if you think you're storing your honey okay, or if it crystallizes for some reason. That's not a key indication of why. I can't tell you why it's crystallizing, but the amylase activity can be an indicator that it's maybe being stored too warm, too cool, some, you know what I mean? Like it's kind of a
Interesting.
again, I've always read it as a freshness evaluator. I'm still learning about that one. Now the protein lipid, um the protein lipid for the pollen, that one we have pretty much up and ready. I'm just dealing with like red tape and bureaucracy.
So why
And
would
then
somebody want to know that? Like what would be the
so
benefit for like a backyard, or is there a benefit to the
yeah,
backyard?
there would be. And again, I can't speak. I'm not a bee nutritionist. Actually, I can let you know it's not, like I can get you in touch with other people who are. But if hives aren't doing terribly well and you've ruled out some of the other primary causes, it could be
Okay.
that they're feeding on poor pollen quality or that poor pollen quality, poor that pollen of poor quality.
There you go.
Sorry about that. But yeah, because like it's not just what they're eating, it's sometimes when they're eating it. From what I understand, like you want a higher protein pollen more towards the end of the season to get them to overwinter. But you want a higher lipid. I mean, again, I could be totally misrepresenting this. But sometimes I believe like if you want higher lipid in the spring just to kind of get them up and moving.
Okay.
But yeah, it can tell you something about the overall, like, I mean, if you don't, the hive's not failing, but it's not producing as well as you think.
That
That
makes
would be
sense.
one
Yeah.
reason I've had people want to send me samples.
And then this sorry, and then we'll get to Donna's question. The sucrose fructose glucose ratio or presence, what would that tell you other than,
It's
the
more of a nutritional assessment.
it is. Okay. Okay.
Yeah, it's more of a like if you like if people want to be able to say that on a label just from a nutrition standpoint.
Okay. Cool. Okay. So I'll read Donna's question that she typed in the
Did you
chat.
read it?
Because I can now. All right. So Donna says, when bees bring in pollen, we see many different colors, ranging from yellow to orange to red and purple. If you are Carissa's, oh, purple if you're Carissa's bees. My bees are bringing in purple pollen. Is that what you
That
meant?
isn't that what you did? You send us a picture,
Oh,
or,
I don't remember
or
what I
was
did
it
this morning.
blue or purple or something? It was
I can tell you,
Oh,
I
it
have
was
a
blue. Yes, it
yeah,
was.
I know what
blue.
it is. I could hands down tell you that's one of the weirdos that I see a lot.
What is it?
Viper's blue
Yeah, what
goss.
is it?
It's a common roadside weed and they love it. It's related to borage, the herb borage. Borage
Oh,
has
Ah.
pink
borage.
pollen.
I have
Borage
that
has pink pollen.
planted.
I
Okay.
didn't know that either.
Oh, and then, but I guess the point of Donna's Donna is asking, why is the pollen on her cars green, so ugly? The green pollen. That's from pine, isn't it? Pine trees or something?
I can almost guarantee you the greeny yellows are often pine or my maple tree out front, like that thing like destroys my car. Like it's insane. I'm like, really? Really? Yeah, like the tree pollen is almost always going to be like a yellow or a green.
Hmm.
Boring.
And I
Yeah.
only just, believe it or not, I have a fantastic poster that actually shows me the color of pollen and what type of honey it ends up making.
Oh,
Oh,
neat. I would love to
Yeah,
see
cool.
that.
I can find, I have a physical copy of it. When I tried to like find the online image, they were like pound sand because it had the restricted. I was like, okay. But I can forward that along. It's really cool. That's actually how I learned about like blue pollen's a thing. Seriously. I had somebody who had amethyst honey this year. Prettiest honey I've ever seen.
Oh,
Oh, wow.
wow.
It was kudzu and they were foraging on dropped blueberries. It was a combination of both.
Oh,
That's
wow.
really cool.
It was like it was so pretty. I was like, I would totally eat this. But then I had one that was blood red that freaked out the beekeeper,
Yeah.
and it turned out that that was knotweed, which it was like almost predominantly knotweed and goldenrod. I've never seen that combination do that though. It's always just been
Oh.
like dark brown.
I've
It
had
was
some red
crazy.
pollen, but not it, but it was the spring, not in the fall.
And
Those are
this
fall.
was
Yeah.
like blood red. It wasn't opaque. It was a very clear honey, but it was red.
Oh, the
It
honey was red.
literally, yeah, it looked like you
Oh,
added Kool-Aid and turned it into honey.
Oh,
interesting.
wow. I thought she meant pollen too.
Yeah.
Mm-mm. Nope.
Approximately how many honey samples do you receive annually from PA alone?
Wow, I would say I'll do it by a percentage. So it's the lab has only been accepting samples since 2023. The first year I had about 1,500. Last year I had over 3,000. And that's a
Wow.
combination of research and beekeepers. Okay. And before I forget, anybody in your group who wants to submit samples, everybody gets the discounted rate because you had me come invite me and give me a talk. All you have to tell
Yay!
me is you're part of the Chester County group and make sure I give you the discounted rate. But yeah, it blasted to like next level last year. And by the way, this is a part-time job. This is a 20-hour a week thing. That's all Penn State wants the lab be open. I'm like,
Oh, wow. Oh,
I have two technicians, but we're all part-time. So if it takes me time to answer an email, that's why. But yeah, and also if anything gets screwed up, you can almost 99% blame me for it because I do most of it. But yeah, we doubled last year. Pennsylvania probably makes up anywhere between 5 to 15. The beginning, Pennsylvania was like 80 to 90, but I have every state. I have 7 countries, and at least 3 continents now that
oh
I've
my
done.
gosh. Wow.
My favorite was Fiji. I had mangrove. I was like, what? It was actually kind of
Cool.
fun because I looked at all the plant genus that were there and I was like, I have no idea what any of these are. And it's amazing. Hawaii is pretty fun too. Although
I
you're gonna
use
laugh, but the favorite plant in Hawaii is still clover. They still love clover even in Hawaii.
Oh my gosh.
I used the service for the first time last year, and I was amazed at everything that was in mine. I sent
I
in three
recognized
samples.
your name. I
Thank
recognized
you. I recognized your name when I saw it in my email today.
your name.
Yeah, I
But
love.
I was really afraid because I had a super bloom of mountain laurel on top of the mountain. I live near the highest point in Pennsylvania, which is Mount Davis, and they had a
Nice.
super bloom of mountain laurel up there, and I was really concerned about that honey. I didn't want to sell honey that had a lot of mountain laurel in it, and it
Hmm.
was. I left that on the hive the whole year, and it did get diluted down, but it still was 34% mountain laurel whenever she analyzed it.
Oh,
Yeah.
wow. Does it
I,
taste bad or something? Is that why you didn't want?
Well, mountain laurel honey is supposed to be poisonous, toxic.
Oh, is it? Oh my gosh.
That's what they used to
I've heard
call
that
mad
about
honey.
rhododendrons. Yeah, rhododendrons, rhodies and
Oh,
azaleas.
mad honey. Okay.
I
Okay.
But
always
it's
wonder
Yes.
though if it's really like the plant tox is probably super diluted. It would be interesting to ask the folks at Cornell if they've ever seen that. They do testing for toxicology and stuff.
Interesting.
I was really afraid of it. If I would have taken that super off, they filled it up in like a week and a half in June, and I got concerned about it. And I had already put another super on. If I'd have just taken that one off and put the other one on, that one would have probably been a higher percentage because nothing else
Interesting.
was blooming up there in that area except mountain laurel. Um,
Interesting.
it doesn't have a bad taste. It's supposed to be real bitter if it's pure mountain laurel, and it doesn't have a bad taste. But I've been using that to feed back to my bees when I need to give them some feed. I just didn't
Yeah.
want to sell any bad honey to somebody. But the
And
other
Yeah.
I
two
will say,
samples were fantastic.
nice. Well, I'm glad to hear. Yay! It's funny because like I've oddly enough sometimes the early spring honeys in general that are predominantly like the tree pollen, I get a little bit of a medicinal back flavor to it.
Hmm.
It's not repulsive. I mean, I certainly would have no problem putting it in my tea. Now, spotted lanternfly honey, that would be going into baked beans, not my tea.
If
Baby.
you sell honey to somebody, if you sell honey to somebody and show them what all is in it, that really impresses them too.
Those wheels, yeah, I use those, Roger. Like, yeah, the pie charts,
Yep.
they're
If you
great.
show that, a lot of people took pictures of it after they bought the honey.
Although my one said oak, like that was the top percentage, and I was like, wait a second, oak doesn't have any nectar, does it? I don't think it does. So, but it was in my honey.
Well, the
Yeah,
pollen would be there, not the nectar. It would be the pollen.
Yeah, right. But it's like you would assume that the whatever the percentage of those pie charts are, they're visiting that tree for their nectar as
Mm.
well, and that's why the pollen's in there.
there's so much pollen in early spring trees. Okay, so my joke for a while, I don't know if anybody's familiar with Peppa Pig and the glitter episode, but like my kid, like it literally shows the teacher in this Peppa Pig episode like bringing the tube of glitter out like it's like a nuclear reactor. Maple and willow and oak are the glitter of the pollen world. They will show up the rest of the year no
Is
matter
that
what
right?
else the bees went to. They collect so much of it and it gets spread around the hive. I mean, it's not like they run through there and vacuum it in winter, you know. I mean, I barely, if
Right,
I didn't
right.
have a robo vacuum, God knows what I find on my floor.
Funny, funny.
Well,
Okay.
thank you very much, Michelle, for the rates that you give us and the diagnostics. Like I said, they were
Thank you so much
fantastic.
for having me too. I love talking to people.
Yeah, I
I
like.
live nowhere near these guys. They're clear at the other end of the state.
I
And
wondered, I was like, Roger, like, aren't you kind of more on my
when they
end?
had their conference, they wouldn't deliver my meal either when they
Oh,
did their online
sorry.
conference.
Uber doesn't go that far, I guess.
No.
Uber Eats. Okay, I don't see any other questions. I guess I have one about the spotter and lanternfly analysis. Do you envision expanding that in terms of, I mean, of course, I don't really know how it's done, but like, do you anticipate like expanding the results to more of a presence absence kind of thing? Or
Potentially, it's so hard because it's almost impossible to tell if it's from one way. Like, and the other thing is, you have to look at size. The nymphs make a lot of honeydew, but they're so much smaller than the adults. It would be really difficult to equate it to, did it feed the bees forage on a group of like nymphs on a tree, or just one or two lanternflies they found randomly?
I see. So when you say one bug or like many, it's there, but you just don't know to what extent. Is it like
Yeah, and I
90% or
yes,
okay.
exactly. I would almost say like it's a right now it's reported as a low, medium, high.
Okay.
Quantification again is almost impossible because we weren't there, we didn't see what happened. Really what it's telling you is that you have an idea of that a large percentage of what went into the nectar may have consisted of honeydew.
Okay.
And I can't give a number to that because we don't know how to equate that yet. And
That
I don't
makes
know if we
sense.
really ever will. Like for most research purposes, honey is a monitoring tool. So honey showed up in a couple states, or excuse me, SLF showed up in honey in a couple states before they ever saw a lanternfly, but they knew it was there because the bees found the lanternflies before the people did.
Yeah. Yep. Yeah. Okay. My bees, I have bees out in Reading as you might remember, and they love that lanternfly honey or honeydew, I guess.
Yep.
But there, well, I also don't get a fall bloom out there. I mean, I do, but they don't bring a lot in, so they go
Yeah.
for the lanternfly stuff. So yeah.
Hey,
Howard, do you have a
Michelle.
question?
Yeah, I was going to point out that I have some jars of honey from the 60s and 70s. So if you ever hear of anybody that wants to use them for research for some reason, I'd love to donate them to somebody.
You know what? I might take you up on that. Let me, all right. Yeah, because we're looking to do some we're looking for like older samples that we could do some testing specifically with that amylase dietase test for freshness because that would be interesting. Okay.
Be cool. Yeah. Howard
Um
brought some in and we tasted it and it was really
That
disgusting.
was from like 56, I think it was.
Oh, really? Okay.
sounds like wine that your friend finds in their basement. They're like,
Right.
"Hey, let's check this out."
I have some of those 1988 bottles of honey from that lady from that thing that George and I cleaned out.
Oh, yeah, I remember that.
Oh,
Yeah,
yeah.
I think it tastes like butterscotch, honestly.
Oh.
Interesting.
So yeah, I could donate some of those if you wanted, Michelle.
Yeah, I would take you guys up on that because if we have some older samples too. And honestly, like, and seriously, if you donate, throw some. And also, the other thing before I forget, if you guys have any, if you know of any like schools, community gardens, if you guys have like exhibition apiaries, anything like that, like community outreach. If anybody has samples like that, I run those usually pro bono. I don't charge for education usually for
Nice.
samples. I just did the elementary school my kid goes to. I just ran their samples. We're like, that's so fun. I'm like, and it looks like Bellefonte.
Okay.
Will you be at the Penn State picnic in July?
I'm not sure. I kind of like hop in and out of some of the beekeeper activities things just because like I'm a lab nerd. And I'm a plant scientist, so it's like if you want to ask me beekeeping questions, I'm the last person you probably want to take advice from. But I may. I know it's on my radar. I know we have visiting scientists coming around that time too, so it's kind of like shuffling things around. And the lab has got more limited hours in summer because I'm generally not as busy. I usually do most of my business in fall when people are doing their fall harvests.
Now, how
Uh,
about if I take my samples up and give them to Robin?
Oh yeah, if you do that and have her pass along to me, she'll just leave them. I have to say my favorite name of our bee research. It's the Bee Arthropod Research Facility. What's the acronym? BARF. Best name ever. Robin will leave the samples at the bar for me, and I will happily come get them.
uh,
I drive past it every day to work.
One last question. So Kate says there's a honey apiary that sells lanternfly honey at a local flea market as collected lanternfly poop from bees. Would that honey be an issue? I don't quite understand. I don't quite understand what would be, what's the issue? An issue for what? Kate, if you can come off
I
mute.
guess because well, they think it's poop, but that
Oh.
it's urine, that it's coming from the insect, and then I would think as it's being processed, any things that other than it being repulsive thought wise, that it wouldn't be harmful.
No,
No.
as far as I know, it actually has higher medicinal value. I can't speak to that. Robin Underwood would be a
Interesting.
much better person. She collects the samples and I run them, so she's got a whole project that's looking at that.
I mean, I eat it every day on my yogurt and I'm still alive.
Yeah, I mean, like I
Good.
get the really dark samples. The really dark samples, they almost have like a buckwheat, like that almost like caramelized burnt. I like it, but I'd more like want it in maybe Earl Grey would be an okay tea, but like I'm thinking more baked beans than I am like, like clover honey in my tea.
Mhm.
But no, it's not, as far as I know, like I don't I usually tell people there is no reason to be afraid of it. Like you don't even have to, you're not required by the state to report that it's, lanternfly.
Right.
So no, I wouldn't. I don't think that there's any. I mean, if people want to, I mean, what was it? The one guy was selling Honey of Doom. I thought that was hilarious.
Oh,
Like
Doomlin,
he
yeah.
marketed it as like lanternfly, and I'm like, okay,
I'll
but
have
yeah,
to let
I
them
don't.
know that it's not really poop.
Yeah,
No,
it's
it's
not
like it's just what they
like
excrete, right, Michelle?
enzymes. Yeah, it's an ex.
Yeah.
It's a yeah. But I also laugh because my daughter was like, ugh, and I was like, yeah, but what's honey? And she's like, oh, that's right. And I'm like, it's like start.
Yeah.
It's a little bit different.
Right. Thank you.
You're welcome.
I
Okay,
have one more
go ahead,
question.
Ann.
Can you hear me?
Yes.
Yes.
Yeah. I've heard of honeydew being created from linden trees. Have you gotten any of that honey?
I cannot specifically test for aphids, although I've heard the same thing. The only time this is kind of more this is more like we think this is what's happening. We'll have things like pine. I've had pine, I've had different basically pine's the big one, but aphids often will feed on young pine frond or pine, like when you're starting to get new growth on a pine tree, and that'll show up in a honey sample if the bees forage on aphids. I can't specifically test for aphids themselves. We usually kind of get it more anecdotally, but like that's not a plant you would expect to be in a honey or pollen sample, and we know it's not our database being screwy, so. But yeah, I have not had one I can specifically pinpoint that, but I can say I'm pretty sure it was. And this was in Florida. It seems that I see a lot. More than once I've had Florida, and then California. I've seen that.
Thank you.
Okay, and that's a wrap.
Thank you very much.
Okay.
Yes, thank you
Yes,
guys for
thank
having me,
you.
and I'm gonna go
That was
walk my
fun.
dog.
That was a lot of fun. Thank you.
Yes. Thank you.
You're
Thank
most
you very
welcome.
much.
Hopefully, I didn't drive you crazy. I try not to make it excessively scientific.
It was great.
I
I
still
thought
got
it was
enough of
great.
work.
Yep. It was
All right,
perfect.
well, you folks have a great evening.
All right, thanks, Michelle. You
Thank
too.
you.
Good night, everybody.
Good night.
If
Bye-bye.
you have more questions, email me.
Will
Good.
do.
Awesome.
All right, bye.
Bye-bye.